Good afternoon to everyone. I welcome all of you to the 90th lecture in the lecture series in Nonlinear Dynamics conducted by the Department of Nonlinear Dynamics, Bharathidasan University with the support from USA 2.0. It is my pleasure to introduce today's speaker, Dr. Prashant Kumar Srivastava from IIT Patna. Dr. Prashant obtained his PhD from IIT Kanpur and then he did his postdoctoral studies at the same institute. He served as an assistant professor in BITS, Birla Institute of Technology and Science, Pilani, Rajasthan for a few months. And then he moved to IIT Patna as an assistant professor in the year 2011. He has received Best Tutor Award from the director IIT Kanpur and also got Best Teacher Award of IIT Patna in the year 2013. He has several notable publications in his area of expertise and currently supervising a couple of PhD students. His research area includes mathematical modeling in ecology and epidemiology, applications of differential equations in biology, stability and bifurcation, mathematical modeling of HIV dynamics, to name a few. With this short introduction, now I invite Dr. Srivastava to deliver his lecture. Over to you, Dr. Srivastava.
Thank you. Thank you very much, Professor Senthil Velan, for very nice introduction. And first of all, I would like to thank you for inviting me to this series. I learned that there has already been 89 lectures in this series. So thank you for inviting me to this series. I also thank to all the persons who are behind this, behind the organization of this series. I begin with the this talk on nonlinear dynamical behavior of an infectious disease model, which is basically there are various factors which will be considered here in this. One is effect of information, saturated treatment, incubation delay. So these are the three factors which I will be considering. I will go through them one by one. I hope that it will be clear to all of you once I start. So before I go through the main topic, I want to give a little bit of background. And this background is about mathematical modeling. And we all have seen the importance of mathematical modeling recently during the COVID-19 period. So I have mentioned a few points here, which I would like to emphasize. One among them is the importance of mathematical modeling in real world problems is undisputed as far as the sciences are concerned, or even the humanities, you can see. And recently, if we see, this has emerged as a very important tool in almost all areas of engineering, applied science, be it social science, as I mentioned, like humanities, economy, management, etc. Everywhere you will see the use of mathematical modeling. Why is it so? The question arises why this area is so important. The reason being that the alternative mechanism can be explored through the modeling, and we can have a better understanding of the system if we have a mathematical model, because it gives us a robust information about the system. These models which we develop during the process of mathematical modelings, they can be further used for assessing the information about the system without essentially depending on the costly and ethically impossible, sometimes ethically impossible experiments. One of the examples which we have seen recently was about COVID-19 drug delivery, for example, the vaccination, that how many people should be vaccinated, what should be the gap of the vaccination, etc., and so on and so forth. Other such examples are like atomic explosion or the nuclear explosion, and then you want to calculate the energy, how much energy is released when there is a nuclear explosion. So these are certain experiments which you cannot conduct because these are not ethically correct, and sometimes these are costly experiments. Also, you cannot conduct these experiments off and on, and therefore mathematical modeling in such cases helps us a lot. What do these models do? They help us in getting the quantitative predictions, which means in concrete terms, they give us numbers, are the qualitative insights, which means they also provide us if we need just the way the patterns are arising or the way the system is behaving. So we can get both or either or whatever is required from the system. The one kind of model which I will discuss in this particular talk is they will be the theoretical models, and they have their own importance over the empirical models, because in sciences we just not need to understand the patterns which do arise, but we also need to answer why do these patterns arise, which means that we are more into the causation than correlation. Coming back to the core of this talk, which is about infectious diseases. So we have emphasized that there is a great deal of advantage of using mathematical modeling. What about the biological system or the infectious diseases? Infectious diseases usually they are caused by we call them pathogenic microorganism, which means they are very small entity which causes infectious diseases, and these include bacteria, viruses, parasites, and fungi. There are very interesting stories. A couple of them I will tell or I will share with you. For example, we all know now that COVID-19 is spread by virus. HIV is also one such disease. Influenza, SARS, and measles. These are some diseases which are spread through the parasites which are known as through the pathogens which are known as viruses. Bacteria, on the other hand, give rise to diseases like tuberculosis, anthrax, salmonella, chlamydia, and cholera. And protozoa give rise to the diseases such as malaria, trypanosomiasis, or in other words, it is also known as sleeping sickness. I would like to share the stories about two diseases here. One among them is cholera. This was a very, you can say, fatal disease in the last century, and when it was not known, now everybody knows that how cholera spreads, and it spreads via the contaminated food or water. And when people did not know that how it spreads, then usually it was considered that it is some kind of demonly act and ghostly act or something like that. And it was John Snow in the London city in around 1900 who very first understood that the cholera is spreading because of the water. If some of you get time, you just look at the YouTube videos of John Snow and London city. You put the London city, and there is a broad street where this particular water pump was there, where this contaminated water, basically it was connected, the water, the drinking water pipeline was basically getting contaminated with the sewage pipeline, which was also passing through that point in the broad street. So if you just Google these terms, you will find that there are certain videos on YouTube, and then you can see the interesting story how it was understood that how this particular disease spreads. Malaria, I think some of you might be aware that it was discovered by Sir Ronald Ross, who was in India at that time as a medical practitioner, and he was assigned the duty by the British in India at that time. And during that time, it was the region of the Sikandrabad, which is near to Hyderabad. And he was doing his study and trying to understand that how malaria is spreading. And when he dissected a mosquito, I believe it was around 1895, 97 somewhere. And he was dissecting a mosquito which was belonging to a particular species, which is anopheline, and he saw that there is a protozoa which is Plasmodium, and which is causing the malaria, which he found that that mosquito has bitten and was actually infected by that infected individual with the malaria. So why I'm telling these stories, I mean, this has nothing to do with the nonlinear dynamics. However, this has everything to do with the system. And in order to model the system effectively and in order to get the outcome from the system effectively, we need to understand how a disease is spread, or we need to understand how everything works, because then only we can put the things in proper perspective and we can get the model equations, which are mathematical equations eventually. So emphasizing on that point that disease can spread directly or indirectly from one person to another via, for example, in case of measles, this is via respiratory droplets, body secretion in case of chlamydia or HIV, biting of a fly, which is CC fly in case of trypanosomiasis, or biting of mosquitoes in case of malaria, dengue, etc., or ingestion of contaminated food or water in case of cholera. So these are the routes of the disease through which an individual gets infected. Depending on the mode of transmission and fatality, usually the severity of the disease is assessed, and usually an in-depth understanding of the pathogen and epidemiology of the disease helps in control or eradicate it, because then only we can suggest that what control measures should be taken and how we should either control the disease or we can even eradicate the disease. So what is the basic modeling framework if you want to have it? So moving forward, we are getting into the mathematical modeling of the system. So we understand how the disease is spreading. And we also now want to understand that how we can model or how we can convert the system, physical system, into the mathematical equations. So usually this basic model framework works that you divide the entire population into certain compartments and you see that how the flows into the compartments or out of the compartments are happening. So we consider that the total population is NT, and we divide this total population into certain compartments depending on the status of the disease. First one is ST, which is referred as susceptible population. That means this contains those individuals who are healthy, not yet infected with the disease, and are prone to the infection, or they can catch the disease. ET represents those individuals who are exposed to population. That means they are exposed to the pathogen or to the disease. However, these people or these individuals, they are not capable of transmitting the disease to another individual. So they are carrying the pathogen, but they are not, at the same time, they are not able to transmit the disease. Infective individuals, IT, represented by IT, are those who are infected as well as they are capable of infecting others. So we call them infected as well as infectious. And recovered individuals are those individuals who are, who had disease in the past, and then either by self-immunity or by treatment, they got cured, and therefore they became immune. Now, naturally, certain diseases offer immunity which is permanent, which means that the individual who has who has got the infection by the disease will not get this infection by the same disease again, or this may be a temporary immunity, which means that the individual may get the infection once again. In terms of modeling, whether the recovery is permanent or temporary, sometimes also depends on the modeling time period. I mean, what I want to say here is that if you want to consider that the individuals are losing the immunity, however, the modeling time period is, let us say, 1 year, and individuals are losing immunity after 2 years, then there is no point of considering that they are losing immunity, because it's essentially the time period is larger than the modeling time period, and therefore it's less probable for the individuals to return back to the healthy class, or the class where they are prone to infected. So with these compartments, what we do is that we write the rate equations, which means the rate of change of population. And if you see the first equation, it says that the rate of change of the healthy population equals to the inflow rate of the healthy population, which is f of s, minus the outflow. The outflow is happening by the interaction of the healthy individuals with the infected individuals. Therefore, healthy individuals are becoming infected and they are joining the exposed class. And then this DS is the rate via which the healthy individuals are dying. Naturally, they are dying. So this whole equation translates into the rate equation of the change of healthy population, that is inflow minus outflow. This inflow term, essentially written on the bottom of the slide, is referred as natural inflow rate of the healthy individual or the susceptible individuals. And most of the time it is taken either as a constant inflow, again depending on the modeling, what kind of modeling you are considering. If you are considering very short time period, then you can consider that the inflow rate is constant, because then the population may not be playing an important role. However, if you are considering a longer time period, then you should consider that it is proportional to the total population, because as many as individuals are present, then the inflow rate will be dependent on that. Or even it can be considered as this kind of term, which we usually refer as logistic term. This refers that essentially it is dependent on the healthy population. However, it might get to saturate it to a point where it will reach to a level of the population. Then in the similar fashion, we have written the equation for the exposed class, which means that now that individual have got exposed to the disease, so they have joined the exposed class, and epsilon is the rate through which this exposed individual are becoming infected. And therefore, they are joining the infected class at the rate epsilon E. Delta is disease-related death rate, and therefore these individuals are dying out. And gamma is the recovery rate, which means that the gamma I individual are recovering in the unit time, and they are joining the recovered class. And D here is again the death rate. So this is the general framework in which an SEIR model is considered mathematically. So moving forward, what we have considered in this particular presentation, we have considered two models. One is an SEIR model, where we have considered the impact of, I will mention it, impact of information, which means that individuals have the information about the spread of the disease. And when they have the information of the spread of the disease, they use protective measures. That means they have the behavioral change. And because of the behavioral change, what happens is that this particular term gets impacted. Instead of beta SI term, this changes to something else, which I will discuss later. And further, what happens is that we can also reduce this E class. That means we can remove this E class, and we can consider a delay in the terms here, beta SI term here somewhere. And then we can say that this time period which is spent by an infected individual during this incubation, we call it incubation, means the person who is infected becoming infectious. So that time period when the pathogen is developing inside the body of an individual, that is referred as usually incubation period. So we can consider incubation delay, and using that delay, we can say that those individuals are joining directly the I class. So these are the two important factors which we will consider in a single model. And that's how this talk will be divided into two parts. In part first, we'll consider SEIR model with certain modification. And in second part, we will consider an SIR model with delay. So both the works are actually published, and one of them is published in Chaos, which is an interdisciplinary journal of nonlinear science. And the other work is published in Journal of Applied Mathematics and Computing. So one can refer to these two papers. I can share later on these slides if one is interested to. So the first one is an SEIR model. Here, as I mentioned earlier, we are considering the incidence rate, which is in general framework, we mentioned the incidence rate as beta SI, which is both unsaturated and monotonic incidence rate. So instead of considering the monotonic, we are considering here the non-monotonic unsaturated incidence rate, which is beta SI over 1 plus MI squared. So why we are considering this? Because this essentially says that as the infected population increases, the infection rate increases, and eventually it reaches to the maximum, and then it declines. which says that this happens because there is a behavioral change of the individuals, and they are sensitive towards the information level which is available there in the population about the disease, and therefore they change their behavior, and therefore the infectivity of the disease does not remain the same as it was in the beginning of the disease spread. Further, we have considered the treatment rate here. The treatment is, in earlier case, it was natural recovery only, or the linear treatment, which means that you are able to provide treatment to everyone. However, in the reality, the treatment cannot be provided to everyone. Therefore, a non-linear function, alpha I over 1 plus mu I, this is considered here. And the non-linearity, if you can see, is appearing via this mu parameter. If you take mu is equals to 0, this becomes linear. So this is the nonlinear treatment rate which we are considering. As I mentioned earlier, this is an SEIR, therefore exposed compartment is considered explicitly. The objective of this particular study is to understand the impact of limited treatment availability, which means the effect of this nonlinear function in presence of information about this disease, which means this incidence rate function. And then later on, we will modify this model by removing this E class and introducing the delay, the discrete incubation delay tau we will introduce. Both other factors remain the same. And our objective will be to study the impact of time delay, which is coming via this incubation period. And then limited treatment, which is coming via saturated treatment function in presence of information about the disease. So this is our model. This is our ODE model system, where this information-induced term is considered, which is which I already mentioned, and this treatment term is considered. And if you look carefully, this I can ignore the last equation in my analysis because the rest of the system does not directly is not directly influenced by the R compartment, and therefore it will be sufficient to study SEI mathematically, and R can be known once we know the I, and therefore in the rest of the discussion we will only consider SEI system. So the basic feasible reason for this SEI system is this gamma 1, which is bounded by these terms here. And this is the whole system, SEIR system, so one can easily see that. And also there is an important quantity in case of infectious disease, which is known as basic reproduction number, which essentially reflects that how many individuals are being replaced by one infected individual during its lifetime. So if it is larger than 1, then we say that disease will spread, and if it is smaller than 1, then we usually say that the disease will die out. So this is the basic reproduction number for our model system 1. And on the basis of this model system, we have found we do the typical stability analysis, and then we will go to the numerical simulation. So we obtain that there are two disease-free equilibria, E0 and E1. This is basically trivial equilibria 0, 0, 0. This is mathematically meaningful. However, biologically it does not make any sense because all the population going 0 does not make any sense. And this is a meaningful equilibria E1 K0, 0, which means that there will be no infection at all, and only healthy population will survive. So both these equilibria always exist. And then there is a possibility of endemic equilibria. By endemic, I mean the equilibrium which contains disease, which means that it has E and I both population. So the component of these are given by these factors, and the I expression is a non-linear expression, and the roots of these non-linear expressions are given in the sign of the roots of these non-linear expression are given in the table, and they depend on the coefficients which are given here on the right side. One can easily see that A5 is always positive, A4 is always positive, and A0 will be positive or negative depending on R0 is bigger than or less than 1. So this table says that A5 is always positive, A4 is always positive, and A4 is positive if R0 is less than 1, and it is negative if R0 is bigger than 1. And rest of A3, A2, and A1, they have the sign changes depending on R0 is less than 1 or bigger than 1, and their composition of the coefficients. So in this case, we see that if R0 is less than 1, then also there is a possibility of endemic equilibria. We refer it to as backward bifurcation, which I will discuss later. And when R0 is bigger than 1, there is a possibility of either is unique endemic equilibria or multiple endemic equilibria. So, so the cases are discussed here briefly for the stability. E0 is always unstable. E0 is a 0, 0, 0. It is always unstable. These are discussed via the characteristic polynomial, which are given here. E1 is stable when R0 is less than 1 and is unstable when R0 is bigger than 1. I'm just discussing them briefly because I want to discuss more important things here. And E2, which is our endemic equilibria, that will be conditionally stable. So if I star is bigger than this particular number and S star is bigger than this particular number, and M must be non-zero. So if this happens, then we see that it is locally stable. Then we have also found the global stability of E1. E1 is our disease-free equilibrium. And if you see, these are the Lyapunov functions which we have used for that. If you see the condition of the stability of E1, it poses a very stringent condition on the mu. Mu should be less than this quantity and epsilon should be less than this quantity. R0 must be less than 1, of course. And what the mu is basically this saturation constant. As I mentioned earlier, this is the person who is responsible for the non-linearity of the treatment. So it says that mu has to be very small, which means that the treatment availability should be sufficient enough. Then only you can eradicate the disease. It means even being stable means you can possibly eradicate the disease. So you want this mu to be very small, which means that the availability of medical resources should be in abundance. And in the similar fashion, you are also imposing that epsilon should also be small, which means that you also want the incubation period to be very small. So these are the biological implication of these conditions which we have obtained here. Further, we see that what are all possibilities. We note that if mu is larger than this particular quantity, so mu becomes, you can see that mu plays an important role. So if mu is small, then the global stability is guaranteed. However, if mu is not small, which means that mu is large enough such that it is larger than certain other combination of the parameter, we call it mu critical, then there is a possibility of backward bifurcation, which means the disease does not get eradicated even if R0 is less than 1, and that is shown in this figure here by taking a certain example of certain parameters which are given in this table. So the disease does not die out even if R0 is less than 1, and that is the backward bifurcation, which we have established explicitly. However, if mu is small, then it can be easily seen that disease will die out, and only the transcritical bifurcation, which means the disease will survive only if R0 is bigger than 1, which means that an infected individual is replaced by more than one infected individual. Not only this, we have also found the nonlinear phenomena, local nonlinear phenomena, which is referred as Hopf bifurcation. And we saw that the endemic equilibria becomes unstable at beta is equals to this beta critical value, which is under this condition, which is referred as transversality condition. Under this condition, if this is not equals to 0, so if this condition is held, then we find that that there is a Hopf bifurcation. What do you mean by Hopf bifurcation? That there is, the nonlinearity appears, and the oscillatory solutions, the periodic solutions, they appear in the model system. So this is what we have shown here. In this case, you can easily see that there is a emergence of limit cycle as beta crosses its critical value, beta critical. Before that, it was a stable equilibria, and after, as soon as it crosses the beta critical, the oscillation starts emerging around the infected equilibrium. The condition is shown here for two typical points beta is equals to 0.006, which is less than beta critical. Beta critical is 0.8243. So this is 0.006 is less than beta critical. In that case, you can see the red curve oscillates, and then it suddenly what you can say is that coincides or converges to the endemic equilibrium point I star. However, on the other hand, if beta is larger than beta critical, then the oscillations survive. Not only they survive, but they reach to a stable position, and then they keep on oscillating in the population. That means the oscillatory behavior of the population persists. This is the case when there is a unique endemic equilibria which was existing and the bifurcation is happening. We also found the case when there are multiple endemic equilibria in the range of R0 lying between this 2 point something to 7 point something. And in this case, you can see that in this particular range of, or in this particular window, there are 3 endemic equilibria represented by green and red, and then green and red. And the green here represent that these equilibria are stable. The red represent that these equilibria are unstable, and same thing is represented here. What is also very interesting here, which we observed, that on this branch of endemic equilibria, is the stability of the equilibria changes from stable to unstable, and we refer this as E33. And when we plotted the the local trajectories around E33, we found that there is a Hopf bifurcation which exists around the E33. That means that the stability switches from at this point here in case of case of multiple endemic equilibria via Hopf bifurcation. So this is the case of Hopf bifurcation in case of multiple endemic equilibria, and this is also for those who are working in this area, they can understand that this Hopf bifurcation is basically a local phenomena. So this can be seen from here. We have also seen the case of hysteresis, which is very, very critical. And what is happening here, there is a stable equilibria, and there is an unstable equilibria in this window, and then there is a stable equilibria in this window. And what happens that if we change the parameter and keep on increasing the value of parameter, then there is a sudden change in the equilibria, and as we keep on changing the R0, and then the level of the equilibria changes all of a sudden. And similar thing happens if we go backward in the parameter. So this phenomena is referred as hysteresis in nonlinear dynamics. So this is the point when you can also consider it as a bistability, because it divides the entire region into 2 stable equilibria, which is shown in this phase diagram, that some solution trajectories move to one of the equilibria and other solution trajectories move to other equilibria. So this is the case which we have drawn here for this vertical line, where this E31, E32, and E33 are identified here, with E31, E32, and E33 identified in this figure B. This is the case when R0 is less than 1, but there are more than 2 endemic equilibria. There are, in fact, 4 endemic equilibria. 2 of them are stable and 2 of them are unstable, and these are mentioned here. And the phase diagram is mentioned here in these slides, in this phase diagram. So for hysteresis, this is the set of parameter which we have considered. For the set of for the existence of multiple endemic equilibria for R0 less than 1, this is the set of parameter which we have considered. This is very, very peculiar phenomena of a nonlinear system to obtain hysteresis, and it's not very easy to observe, but it reflects in when the model system is having the nonlinearity, then you can find it. So with these, we come to the what is effect of the information, which is reflected by the parameter M, and effect of the saturation, because we have seen that those 2 other parameters which we wanted to understand that how they are impacting the dynamics of the disease. So we see that when what do I mean by system? Although there is nothing meant like system is stable, what do I mean by system is stable here is that when the endemic equilibrium is stable. So in the case when the endemic equilibrium is stable, you can see that increase in the M parameter, which is increase in the sensitivity of the information. The critical population or the endemic population of I decreases. Whereas in case of the oscillatory system, when the endemic equilibrium is unstable, then you can see that the increase in the parameter M reduces the size of the oscillation. So if there is abundance of the information, M parameter is larger, means it reflects that the information impact is higher. So higher impact of information basically works as a control towards the size of the oscillation. The oscillations are reduced. On the other hand, if we see the impact of the saturation in treatment, which means larger the mu, larger the nonlinearity, which means larger the impact of saturated parameters or saturated impact of the treatment. So if we see infective equilibrium is basically stable, then it does not significantly impact too much on the dynamics of the disease. However, if we look at the oscillations case, when the Hopf bifurcation exists, you can see that the larger you can term it as non-linearity, because mu is reflecting by non-linearity. The larger this mu parameter is, which is reflecting larger in the non-linearity, the increase in the oscillations, there is an increase in the oscillations, which means that there is a higher nonlinear behavior in the system, in the model system, if we increase this parameter. So we have certain information about the model system and how these parameters are changed and how the parameters are varied and how this is impacting the dynamics of the disease. In the second part, what we will do is that we will, instead of considering SEIR model, we will now consider the incubation period as the delay, and we will consider this model now. So here you see, instead of SEIR, now this is SIR model. And this is the delay which is introduced in the model system, which we refer it as a discrete time delay. And now the solutions will come from the Banach space, which is referred here as the set of the continuous functions.
No, we are, excuse me, sir. We are not seeing this. We are not seeing that slide.
Oh, okay. So let me.
No, the next page, because we only see the graphs.
Oh,
this delay model we are not seeing.
Could you see the slide number 15?
The graphs which you are talking. Yeah, this, yeah. Delay model we didn't see.
So now I'm coming back to the delay model. So let me just ask the questions now. So these graphs, did you see 12, page number 12?
Yes, 15. Yeah, we are in the page number 15.
13 also you have seen?
Yes, yes. 14, okay.
14 also you have seen, and 15 also you have seen.
Yes, yes.
Now I come to 15. Can you see this slide?
Yeah, 15 we have seen. Yeah.
Now 16. Now I'm on slide number 16.
No, it is not moving.
Okay. So maybe I will unshare and reshare again. Maybe I will turn off my video so that probably if there is a slow network, that might solve the problem. Can you please confirm which slide number you are seeing?
This is 16. Page number 16 we are seeing. Yes. Delay model. Yes. Yes.
Yes. Yes. So, okay. I'm sorry for the, probably there was some slowdown of the network. So now, I mean, till now we have seen the effect of various parameters on the dynamics of disease when we have considered the ordinary differential equation model system. Now what we are going to do is that we are introducing a delay term there, which we refer as incubation delay. And so there is a huge difference between, ordinary differential equation system and the delay differential equation system. ODE is a, typically a finite dimensional system, and delay differential equation is infinite dimensional system, which can be seen from here because the solutions are coming from this space C minus tau 0 to R3 plus, which is a Banach space. It is a set of continuous, space of continuous functions. So, because, the solutions are no longer points now, they are functions now, okay? So for this model system, we have the basic analysis. We find that the basic reproduction number in this case is this R0, which is given by beta k upon delta plus, which is different from the earlier model system, which is obvious because epsilon is not appearing here, which was our incubation period. So this delay model system, when we analyzed for the case of the equilibrium points, we see that still it has two equilibrium points. One is E0, other is E1. And these always exist. We will not be doing much discussion on these points because these are trivial equilibrium. Our focus will be on the endemic equilibria. So this system possesses this endemic equilibrium E star, whose components are given again by S star, R star, and I star from this fifth degree polynomial. Now this fifth degree polynomial is essentially different from the earlier fifth degree polynomial. although the behavior of the terms looks same, because A5 and A4 are still positive, and A0 is again dependent on R0, and this is positive or negative depending on R0 less than 1 or R0 bigger than 1. So there are possibility of multiple endemic equilibria. However, we are not interested in the multiple endemic equilibria with delay. Rather, we are interested at this point in first in the case when there is single endemic equilibria. That means there is a unique endemic equilibria, and rest of the things we will discuss via numerical simulation. So we will come back to that multiple endemic equilibria case, but not with the case of analysis, mathematical analysis. So we note that I think we can ignore this part here. This I can take up during the question answer session if needed. So because this is our disease-free equilibrium, so we are finding that if R0 is less than 1 and the conditions are satisfied, then this is always stable for whatever value of tau we select. We are more concerned about the introduction of delay on the case of endemic equilibria. So we have found the characteristic equation. Now the difference between the earlier characteristic equation and this characteristic equation is that this characteristic equation also contains the term tau. So this makes this entire expression into a transcendental equation instead of the earlier algebraic equation. And we want to study the stability analysis of this expression, and therefore we want the eigenvalues or the roots of this equation in the negative half of the complex plane. And for that purpose, we want to see that exactly where the roots are crossing the imaginary axis. And therefore, we get this kind of expression, and we find the value of critical value of tau star, which says that at this tau is equals to tau star, there is a possibility of change in the roots of the characteristic equation 5, which means that the number of roots which are lying in the negative half plane that may change. And how that is happening, that is given here on the bottom of the slide. So P star lambda tau, that means this characteristic equation equals to 0. This will have first the condition A0. This is the condition A0. If the condition A0 is satisfied, then this will have no purely imaginary root, which means that there is a possibility of the stability being not changing in this case for introduction of the delay. There is a possibility of one purely imaginary root if the condition A1 is satisfied, and there is a possibility of even two purely imaginary roots if the condition A2 is satisfied. So we have discussed this in detail here. So when there is a one when there is one, so you can see that this is written as A4. When there is one delay value at which there is corresponding to which there is one purely imaginary root, then for that we have found the interval 0 to tau 1 star, in which the endemic equilibrium remains stable. And for tau bigger than tau star, there is a Hopf bifurcation theorem which refers this, that as tau becomes tau bigger than tau star, there is a oscillatory solutions which will exist, which means that we have from stability to instability. So stable equilibrium becomes unstable equilibrium. If the condition A3, which means this
excuse me,
yes,
sir. Again,
yes,
the
yes.
slides are not moving, I think.
Oh my.
Which slide number you are talking?
I am at the 19th slide.
Yeah, 19th we are now. Yeah, now we are seeing. Yes.
I think maybe the network is little slow somehow. I don't know.
Yeah, that should be. Yes.
Okay, so I will be little slow while moving the slides, maybe this time. Okay. All right. So I have to refer back to the slide number 18 now. So here we have mentioned these conditions under which this expression, our characteristic equation, will have either 0 or 1 or two endemic, two purely imaginary roots. And depending on these conditions, we have mentioned here that when this endemic equilibria will be, possessing the half bifurcation. The endemic equilibrium is unique. So the case is when A4 condition, that means when tau1 0 is corresponding to single purely imaginary root. So when that exists, then the A4 is satisfied. That means that for tau lying between 0 to tau1 star, there is a stability, and as tau becomes bigger than tau star, there is instability. So stability to unstable, this is what it is written here in this table. However, in case of A5, there are two endemic equilibria. There are two purely imaginary roots, as I have mentioned earlier. So for the interval tau lying between 0 to tau1 star, there will be only stable equilibrium, and also from tau bigger than tau2 star, there will be only stable equilibrium. However, in between tau1 star and tau2 star, since there are two purely imaginary roots, so there will be instability. So we call it, there are two half bifurcations. I will show you numerically, probably then you will be able to understand. First appearing at tau1 star, and the second half bifurcation appears at tau2 star. And that's why we move from stable first half bifurcation, then that will take you to unstable. Second half bifurcation, that will take you back again to stable. So then the stability switch occurs there from stable to unstable, and then further another switch occurs from unstable to stable. This is very, very, very critical phenomena. We call it as endemic bubble. And E star is always unstable for all tau bigger than 0 if these conditions hold, in this case of either A0 or A1 hold. So we now move forward, and I hope these pictures are visible. Please let me know.
Not yet.
Okay, I will wait for that.
Yes, now we are seeing. Yes.
Yeah, so there is definitely slow network. Okay, so this is the case, as I mentioned, when only one purely imaginary root appears, and that's how you see the stable equilibrium and the half bifurcation exists at tau is equals to tau1 0, as I mentioned in my earlier slide. The value of tau1 0 is 14.93. At that point, the oscillation starts, emerging in the around the endemic equilibrium. And this is the phase diagram of the same thing, which is shown here for tau bigger than tau star and tau1 0 star, and for tau less than tau1 0 star. So stable to unstable. So this is the bifurcation phenomena which is happening from stable to unstable. And these are, the closed orbits and the similar behavior which we have mentioned here. So they are being plotted here in the different fashion. And keeping eye on the time, I will move to my next slide. In this case, we are showing the multiple existence of multiple. Again, I think there may be a little bit of time delay.
Yes.
Is it visible now?
No, we are still seeing page number 20.
Yeah, I think these figures are a little heavy, so that might be.
That might be okay.
Since I'm in the middle of the talk, otherwise I would have optimized the PDF.
Yes.
Yeah. So okay. So now you can see that because of the two purely imaginary roots, there are two critical values of tau star, and therefore from stable to oscillatory solutions to stable again. Oscillatory solutions means unstable. So stable to unstable and unstable to stable. So therefore this S to U, U to S. So this is how the bifurcation is happening. And this is the case when we are referring tau is equals to 30 belonging to the interval tau1 star and tau2 star, and tau less than tau1 star. So these are the two graphs. In one case, you can see that we are reaching to the endemic equilibria, and in another case, we are circulating around the endemic equilibria and formulating the periodic oscillation. So this is the explicit time series around the endemic equilibria, which shows that there are oscillations if we are in that tau is equals to 30, which is lying between tau 1 0 and tau 2 0. One can easily see that tau is equals to 30 lies also from the first part of the figure, from figure A, that it is 30 is in between tau 1 0 and tau 2 0. So this I will again wait for a minute. This is very important result, which is the nonlinear phenomena which we have observed, and we have not observed so far such phenomena in infectious disease dynamics. So as I mentioned earlier, that Hopf bifurcation is a local phenomena. So what is happening here? You will see that this is about the multiple endemic equilibria. Please confirm when you see the slides.
Yes, we are seeing page number 22.
Correct. Okay, fine. So in this case, if you see tau is equals to 0, tau is equals to 0 refers that there is no delay in the model system, which means that there is, this is ordinary differential equation system. One can see that tau is equals to 0 is the same as ODE model system. And in that case, there are three endemic equilibria referred as E1 star, E2 star, and E3 star. However, in this case of E1 star is stable, E2 star is unstable, and E3 star is stable. So this is bistability. So one can understand that this is a bistability, right? So this is bistability in case of OD model system or in absence of delay. Now what happens when we introduce delay? You can see from this figure B, there is a coexistence of two Hopf bifurcation, although those Hopf bifurcations start bifurcating at two different time points. For example, in this case, the Hopf bifurcation bifurcates around tau is equals to around 11 and half, and in this case, this bifurcates around 14 and half. So the exact values are mentioned here, 12.36 and 14.33. So those are the exact values at which point these bifurcations are appearing. So what is happening here, that the model, when it was showing the stability of two equilibria, both the stable equilibria start showing the Hopf bifurcation at different time point. Comparing to the point, because these values are, you see, at tau is equals to 13. Now you see that the some solution trajectories are going towards the E1 star, which is showing the oscillatory behavior, because E1 star is essentially showing the Hopf bifurcation. E1 star is the first one. It is showing the Hopf bifurcation, and therefore these solutions are converging to the oscillatory solution. On the other hand, if we start near the E2 star, you see the solution trajectories are moving to the E3 star. So in this case, there is a bistability, which is very different bistability than the earlier bistability. And further, when we consider the tau values in this range of 20, which means that when both the endemic equilibria are showing Hopf bifurcation, then the different points take us to different oscillatory solutions or different periodic solutions. So in case of bistability, what happens that we reach to different points. However, in this case, there is a bistability of the periodic solutions. So we reach to two different oscillatory solutions when we start from two different initial conditions. So it's a very different bistability than the usual bistability which we understand. Although it is also bistability in the exact sense of how we define the bistability, that you move from two different initial conditions and you converge to two different solutions points. And this is what we have then again tried to check our model system, that how it behaves when we vary the parameters beta, which was infection rate, with respect to tau, when we vary the parameter m, which is the effect of information with respect to tau. And we see that in case of when we vary the parameter beta, there is no stability there is no multiple stability switch.
Excuse me, sir. I think
yes.
you are talking next. Yes.
Oh, okay. Yes.
Yes, now we are seeing 20.
Okay. So in this case, we have drawn the figures with multiple beta and tau, m and tau. And if you see, this is the black color curve, which says the critical value of the parameter tau, which is responsible for the bifurcation. So if we are below this, then we are stable. If we are above this, we are unstable. So there is only one single stability switch. However, in this case, when you are choosing m as the parameter and m versus tau figure, you can see that there is a a parabolic shaped curve, and then there is a possibility of two switches. So if we draw a line at, let us say, tau is equals to 15 or somewhere anywhere tau above 14, you can see that there will be two stability switches, one from stable to unstable and once again from unstable to stable, and that's what we have shown in the previous figure. So this shows that there is a possibility of multiple endemic equilibria existence in oscillatory form whenever there is a step, whenever there is a change of parameter and change of delay, which is the incubation delay. We have also seen, we have changed some parameters and we wanted to see what is the impact of delays. So one obvious impact was seen here is that if we increase delay, then the size of the oscillation increases as we increase the delay parameter. So these results are mentioned here. So with this, we come to the end of the discussion, and we mention, I hope this slide is visible, to the conclusion of this talk. And the major points I have already covered here.
No, we are not seeing that conclusion.
Yes, hopefully it will be shown. Yeah. So the major points we have already covered here, and we have seen that the disease transmission rate beta, the treatment rate alpha, the treatment parameter mu, and the incubation delay tau. These parameters, they have their own impact on the effect of the disease. And in case of limited treatment, which is reflected via mu, we see that even if R0 is less than 1, basic reflection number is less than 1, then also the endemicity is there and the bistability of the equilibrium is obtained. We have also found the multi-stability, which means we have found the multiple equilibria, which are showing multiple kind of stabilities in this case. And this is interesting, which is the last point, which I have discussed in detail. The bistable behavior of non-oscillatory to bistable oscillatory limit cycles, that is also obtained in this case. And the delay parameter, which is reflected via the incubation period, that causes the existence of bubble or the oscillatory double half bifurcation, or we sometimes refer it forward and backward half bifurcation. So those kind of bifurcations in the model system. So the richness of dynamics is exhibited when we consider the delay in the model system in case of another class of compartment in the model system. So this is the end of the talk. Thank you very much. I shall be happy to take up the questions if there are any.
Thank you, Dr. Srivastava. So I would like to conclude the session by thanking Dr. Prashant Kumar Srivastava for accepting our invitation and giving a very wonderful talk today. So we learned several new insights on SEIR model. Thank you, sir. Thank you very much.
Thank you very much, Professor Selvan, Professor Senthil-Vellen, for inviting me. And it is really wonderful to have interacted with many of you. And I hope to see some of you very soon. So all the best. Thank you.